afternoon, everybody. Thanks for coming. It's been a great week, and we're in the home stretch, so thanks for coming. My name is Amos Gutnick. I'm a business development manager at PackGene. My background is in molecular cell biology and AAV research, academia, pharma, biotech startup. And over the years, I've developed, as I'm sure a lot of you have, a real appreciation for the significance of the roadblocks that are really keeping this amazing technology from many of the patients that could benefit from them the most. So today I'm going to share how PackGene's platforms are making high-quality gene therapy manufacturing more accessible, more cost-effective, and helping to overcome some of those barriers. So PackGene is a leading global CRO and CDMO that's focused on gene therapy. We offer end-to-end services from plasmid construction to AAV manufacturing, supported by over 110,000 square feet facilities, a track record of over 10,000 projects for over 1,000 customers worldwide, from small, scrappy academic labs to big multinational corporations. So since our founding in 2014, we've expanded very rapidly. I think now we're over 30 countries at least. And very recently we launched our new 35,000 square foot GMP site in Houston, Texas. It's fully operational, aligned with global regulatory standards, and is currently producing GMP materials for clinical trials. So our GMP capacity utilizes industry standard single-use bioprocessing, serum-free hex suspension systems to deliver over 1,000 grams of GMP plasmid DNA, over 1E19 viral genomes of GMP AAV annually. And we've already supported dozens of INDs, successful GMP batches that scales up to 200 liters. So for all the transformative potential of gene therapy, I think we can all agree that the cost of manufacturing is still a significant prohibitive bottleneck for development and commercialization of this modality. And at PackGene, we address this by offering scalable, high-quality AAV and plasmid platforms that incorporate innovations at basically every level of our production pipeline in order to increase yields, lower impurities, and thereby reducing the cost per batch. So I'll introduce you today to 3 core platforms that have allowed us to achieve this goal so far. First is the Pi Alpha high yield AAV platform, the Pi Omega high yield plasmid platform, and finally our Pi Icosa capsid engineering platform. Okay, so let's dive into the details of our, I think, flagship innovation, which is the Pi Alpha AAV high yield platform. So the core of Pi Alpha is our proprietary RedCap plasmid, which is engineered with a novel non-coding regulatory element that consistently boosts AAV yields by 3-fold to 8-fold across multiple serotypes. And this enables, obviously, more high-quality material per batch. Our novel plasmid design also sharply reduces capsidated DNA impurities from 3% all the way down to 0.2, 0.1%. And the data shown here shows 3 different gene of interest constructs, and all of them benefit from the same improvement. And that means safer vectors, less risk of off-target effects or regulatory hurdles from residual DNA. Now, beyond plasmid engineering, or more in parallel with our plasmid engineering, we systemically optimized transfection, engineering, harvest parameters, which has resulted in 80–90% full capsid enrichment post-AEX chromatography, which is, again, robust across all serotypes. And our approach really integrates the molecular design with our process development to really maximize yield and product quality. Importantly, Pi Alpha is fully scalable, so we see seamless process transfer from 3 liters all the way to 200 liter batches, and we've achieved single batch yields of 1E17 viral genomes. with residual host cell DNA, again, kept at industry-leading low levels, and that ensures consistent quality as you take your programs from preclinical development all the way to the clinic. So further process optimizations reduced other impurities, encapsulated plasmid DNA levels below 100 nanograms per 1E13VG, maintaining key product quality attributes, and additionally, residual host cell DNA is reduced, again, more than tenfold. And the two figures below are real-world cases of studies where optimized process significantly lowered host cell DNA content. So if you look at the top right, then you're going to see a comparative study, again, reporter gene expression in day 14 mice in material that's produced by a more traditional manufacturing pipeline versus PackGene's process. And you see markedly stronger expression, of course, and in the PackGene process. So this is just some of the innovations in our pipeline. Another important one is engineered optimized monoclonal suspension cell lines that are efficient and well suited for our pipeline, as well as a dual plasmid transfection system that we've developed that improves yield and consistency. So just to describe a little bit our two plasmid system, here you see data that's validated in single-stranded AAV, 9, and it outperforms traditional 3-plasmid process, higher yields, half the residual host cell DNA, and it maintains vector purity. So this simplifies manufacturing, enhances regulatory compliance, and importantly, uses less plasmid. And as you take the program towards the clinic, the costs of manufacturing GMP plasmid are very prohibitive, so this is an important advancement. So now we're talking about manufacturing plasmids. I'll talk a little about Pi Omega, which is our high-yield plasmid production platform. It produces optimized plasmid backbone designs and complements it with improvements to the protocols all through the process. So by engineering plasmid backbones for our GOI plasmid, helper, and RepCap, and by optimizing ITRs, we've achieved 4-fold higher yields and additional 3-fold yields from the ITR modifications. And again, this process is fully scalable. So we know what to expect when we start out in low scales, what to expect as we approach IND. So our 2-step purification process maintains high standards. You can see here, we're only purifying through these 2 columns, but endotoxin-free, high supercoiling, non-detectable host cell protein. But we achieve higher recovery because it's only 2 columns, and even lower residual host cell DNA. So we're reducing costs again without compromising quality. I'll just note here that this improvement in recovery becomes more significant the bigger the plasmid is that you're trying to run through these columns. So it goes hand in hand with our 2-plasmid transfection system, because when you switch to 2 plasmids, the plasmids are much bigger. And so this innovation, again, complements the plasmid design. All right, and finally, beyond manufacturing, our Pi-icosa platform leverages AI-driven capsid engineering to discover novel AAVs. So enhanced tissue targeting, of course, improved safety, and importantly, improved manufacturability. And these advances reduce the required therapeutic dose that you would use, lower toxicity, support safer and more effective gene therapies. So regarding some of the capsids that have come out of the Pi-icosa platform, you might have seen a couple of our posters detailing engineered capsids that dramatically reduce liver exposure, and toxicity, and preserves muscle tropism in human and in mouse models of Duchenne. So AI-assisted discovery enables us to rapidly identify and validate our capsids, precise tissue specificity, and expand our toolbox and really face the next generation of gene therapy, which I think is hopefully right around the corner. If you missed our posters or you want more data about our capsids or any of our processes, then just reach out to us. We're happy to provide you with information, data, answer your questions. So at PackGene, we don't just focus on manufacturing. We really support the entire gene therapy journey from the very beginning. So as your program advances, our Houston GMP facility is ready to support clinical and commercial production. So it's a state-of-the-art facility that has 6 GMP clean rooms, dedicated process and analytical development labs, QC and warehouse infrastructure, and it is, again, fully operational now. So, again, I'll just mention that we are available to answer your questions. Our booth is 1821. We have a lot of information there that you can browse. Some good swag too. And that's it. Thanks for your attention. And please reach out to us. And if you scan this QR code, it's following us on LinkedIn. We actually have a pretty neat weekly digest with all the news that's worth printing in the field. That's also pretty useful. All right, thank you. Thanks a lot for your time.